Introduction to Microbiology. Teaching objectives. To give an overview of the history of microbiology, to introduce the following: the extent of the microbial world, microscopy theory, fundamental and quantitative technique, use bacterial and viral growth characteristics, microbial phylogenetic and metabolic diver. Learning outcomes. At the end of this section, students will have an appreciation of /or be able to. The important developments in microbiology, describe basic and specialized microscopy techniques and their applications. The extent of the microbial world, describe the important differences between prokaryotes, bacteria, anarchaea, and eukaryotes. Learning outcomes. 2. At the end of this section, students should be able to describe basic and specialized techniques for quantifying microbial growth, summarize the process of bacterial and viral reproduction, and describe the dynamics of a bacterial growth curve and the placacy, indicate how bacteria are divided into groups according to their phylogeny /or metabolism. Lecture plan. Scope of microbiology, extent of the microbial world, history of microbiology, techniques, microscopy and staining, pure culture methods, quantitative methods, microbial growth curve, placacy, microbial diversity, phylogeny, substrate utilization. Introduction. Microbiology. The study of microorganisms, organisms too small to be seen with the naked eye except in large groups, effects of large numbers often visible, for example chemical reactions in soil horizons, for example toxin and gas production in incompletely sterilized food cans, for example disease in animals and plants. Microbial world. Virus, bacteria, eubacteria, anarchaebacteria, fungi, yeasts and molds, protozoa, microscopic algae. Benefits. Maintain balance of environment, microbial ecology, basis of food chain, nitrogen fixation, photosynthesis, digestion, synthesis of vitamins, manufacture of food and drink, genetic engineering, synthesis of chemical products, recycling, sewage, bioremediation, use microbes to remove toxins, oil spills, use of microbes to control crop pests, normal microbiota, harmful effects, cause disease, basis for biotourism, food spoilage. Pioneers of microbiology. Robert Hooke, UK, 1665, proposed the cell theory. Observed cork with crude microscopy. Living things are composed of self-spontaneous generation. Some forms of life could arise spontaneously from non-living matter. Francesco Redi, IT, 1668. Redi's experiments first to disprove S.G. Antoni van Leeuwenhoek, Delaware, 1673, first observed live microorganisms, animalcules, Schleiden and Schwann, deformulated cell theory. Cells are the fundamental units of life and carry out all the basic functions of living things. Pasteur, Fr. and Tyndall, UK, 1861, finally disproved S.G. Louis Pasteur, 1822–1895. Chemist. Fermentation, 1857. Pasteurization, heat liquid enough to kill spoilage bacteria, 1864. Vaccine development. Rabies proposed the germ theory of disease. Proposed aseptic techniques, prevent contamination by unwanted microbes. Director of Pasteur Institute, Paris, 1894. Joseph Lister, UK, 1867, used phenol carbolic acid to disinfect wounds. First aseptic technique in surgery. Robert Koch, Delaware, 1876. Postulates. Germ theory, 1876. Identified microbes that caused anthrax, 1876. Tuberculosis, 1882, and cholera, 1883. Developed microbiological media and streak plates for pure culture, 1881. Koch's postulates. The specific causative agent must be found in every case of the disease. The disease organism must be isolated from the lesions of the infected case and maintained in pure culture. The pure culture, inoculated into a susceptible or experimental animal, should produce the symptoms of the disease. The same bacterium should be re-isolated in pure culture from the intentionally infected animal. Branches of microbiology. Bacteriology, study of bacteria. Mycology, study of fungi. Immunology, study of immunotide. Word Jenner, UK, developed vaccination, 1798. Mechnikov, Rue, discovered phagocytes, 1884. Paul Ehrlich, Delaware, theory of immunity, 1890. Virology, study of virus. Gering, Nebraska, discovered intracellular reproduction of TMV, Coined the term virus. 1899. Parasitology, study of protozoa and parasitic worms. Chemotherapy, treatment of disease by using chemical means. Antibiotics, produced naturally. Synthetic drugs. Ehrlich. 1878. Used arsenic compounds to fight disease. Magic bullet. Chemotherapy. Alexander Fleming, Scotland. 1928. Discovered penicillin. Solomon Waxman, Ukraine. 1934. Discovered streptomycin. Problems. Toxicity of drugs equals selective toxicity. Resistance of bacteria to drugs. Recombinant DNA technology. Recombinant genetic engineering / biotechnology. Microbial genetics, mechanism by which microbes inherit genus. Molecular biology, structure and function, expression of genus. Molecular epidemiology / diagnostics, biotechnology. Diagnosis / gems for industrial, pharmaceutical and agricultural applications. Improvements of agriculture, plants and animals. Gene therapy, inserting a missing gene or replacing a defective one in human cells.